Before I get underway, I wanted to say a couple things I forgot to start off at the beginning. One, I caught up on grading. Of course, you guys are just continuing to work. So as of, let's call it 8 o'clock last night, I graded everything that was there. And then I looked at things this morning, and there's some new stuff. Okay, so I'll try to get to that stuff this week. I have to get caught up on my A&P2 classes grading, which is what today's for. We should have a reasonable picture of what your grade looks like right now. When you're looking at your grades, you'll go into the My Grade section of Blackboard. I can't demonstrate what it looks like for you because Blackboard's got this kind of annoying thing that I'm an instructor, so I don't have any grades. Obviously, I'm not taking a class, and it doesn't, show me what things look like to you. If you go into the My Grade section, I believe it'll have it'll have all the assignments listed. I believe if you click on either the assignment name or the score for the assignment, it'll take you to a picture with a little bit more detail about how you did on that. And I think you have to click through again, probably on the name of the assignment, and you'll see even more detail where it should have comments that I've made. So except for auto-corrected questions, I can write it in comments. And if I don't give you full credit on something, I say something to explain why. It might be you haven't answered the question enough depth, or I might explain what the correct answer is, and you're kind of off in left field with yours or something like that. I might not say something specifically about your answer. I might just say what the correct answer is. As I'm grading things, if I have several people that are answering something incorrectly in the same kind of way, I'll usually copy and paste comments from one person to the next. So it might lack a little bit of the specificity for yours, but it does get the point across as to what the question is looking for. So you can do that. Let me just mention this while I'm talking about it. The auto-corrected questions, multiple choice doesn't apply to, obviously, but for any of the fill-in-the-blank ones, spelling counts. I can put in multiple acceptable answers, like I could put I don't know, my go-to example for this, which actually is in the syllabus, is I could put polar or hydrophilic, which are essentially interchangeable terms, or some of the stuff I've talked about today. Dorsal and posterior are interchangeable terms, especially when we're talking about spinal cord structure, that kind of stuff. So I can put those things in, but I'm not going to put in common misspellings. You have to spell things correctly. And I mention that because I was going through, and these have been sitting on my to-do list for a little while. I went through and graded some of the body cavities and directions makeup assignment that some people did. And some people might have done them without having been required to, which is fine. I just grade everything that comes through. But there were 5 or 6 attempts, and this is why I think there are a number of people that didn't have to do it, because there weren't that many people that had to do it. But every single one of the 5 or so attempts I saw on that misspelled the word sagittal. And so every single person that did the makeup assignment did not get the point for sagittal because they didn't spell it right. Yeah. 2 G's, S-A-G-G-I-T-A-L, I think it is. And it was spelled with only 1 G. Or maybe it's 2 T's and it's spelled 1 T. I can't remember offhand what it is. It's one of those words I have to sit down and really think about how to spell it. But when you're submitting answers, be sure that you pay attention to how things are spelled. If you have misspelled it, you will not get credit for it. It will not tell you you didn't get credit because you misspelled it. It just say it's wrong. And you might think that means because you didn't put the right answer in there. Please pay attention to spelling. And hopefully for that particular assignment, a lot of people are doing it above and beyond what was called for, so they're not going to be penalized for misspelling that. But when that kind of stuff happens, it causes a problem. In the skin part 4 assignment, there's a question in there that has always been problematic, and I finally decided how I wanted to fix it, but not in time to help you guys. I reworded the question, and I did it in the system so that I don't forget to do it after the class is over. If you go in and look at this question, it will not look the same way that it did when you took the test. And I'm just letting you know that. I graded it based on how it was answered, and then I went in and I changed it for future iterations in the course. Just because I don't want to mess this up. It's the one that was about glands, and the way that it's worded now, if you haven't completed that assignment, you're going to get the new version of the question. So what I'm about to describe doesn't apply to anybody else. But what it had said in the question was something in effect of list the different types of glands that are associated with the skin. And I gave you the first one. I said sudoriferous gland secretes sweat. And then there are three pairs of blanks after that. The point, and I presented the types of glands in class specifically because I knew this question was coming, was sebaceous glands secrete oil, mammary glands secrete milk, and ceruminous glands secrete wax. I said it specifically that way because that's the way that the question was set up, to accept oil, milk, and wax as answers. If you wrote in sebum instead of oil or cerumen instead of wax, you still get credit for that. But part of the reason why I had to redo the question is because there was no order to the question. You could have put any of those glands in any of those positions. So I had to put all of those as possible answers in. And it just gets really cumbersome when I put lots of extra types of answers in there. So if I put oil, milk, wax, sebum, cerumen in for every single one of the second blank in each pair, the question would have gotten unwieldy. So I wanted to go through and change it. So now the question's worded so that it's specifically looking for only one word. So like it says, the sebaceous gland that secretes blank, comma, an oily substance. So that should be obvious what it is. If you go back and look at that question, I actually don't know if you've already taken it, if it'll show you the rewritten question. And if so, the rewritten question and the answers you put in will not probably correspond. Don't worry about that. I graded it based on the original version of the question. And sometimes in the system, when I change a question, if you've already taken the test, it might show you the old version of the question. But just go back and look at that. That's just to warn you. I changed the question. I graded it based on the way that it was originally written. And pretty much everybody had done that question, that assignment. But there might be a couple of people that haven't gotten around to it. So you're going to see the different version of the thing and get that. Answer it based on that. So, sorry.
It changed the question, but it down here it says exact max, but up there it marked it wrong, but you fixed it. So
Right.
it gave me an 18 out of 18.
Well, 6 out of 6 on that question.
Yeah, but 18, you know.
Yeah, so you'll see it. The score will be based on how it was originally worded when you answered it. I graded everything and I went through and changed it. So if you go back and look at it and you're like, These words obviously don't fit in the sentence. That's because I changed the sentence after I created it for you. So I just wanted to mention that. Okay, so let me continue on here. We were looking at this dorsal root ganglion slide, showing you the different, the unipolar neurons here. I want to go on and show you, where to go, the cerebellum. This is part of the brain. The cerebellum is, the word cerebellum means little brain. When you think of the brain, you probably think of this big wrinkly thing right here. And the big wrinkly part that's making up most of what you see is the cerebrum. There's actually another smaller wrinkly part, which is a separate piece of this model that I'm pulling out here. And that's the cerebellum. Okay? Now the cerebrum and the cerebellum both have this kind of folded outside structure to it. When I was showing you pictures of the brain earlier, I said sort of a cauliflower appearance to the outside of the brain. That's what I'm talking about. So this is the cerebellum, and we're looking at it under some magnification, so we're seeing some detail. But you can kind of see a line right here. This is the edge of the tissue. Out here, we're looking at clear glass, essentially. And then where it comes down into this sort of fold between two pieces of tissue, this would be where we see the wrinkle in the tissue. And it actually wrinkles quite a bit. So this is a fairly deep fold that you can actually see. It just wrapped all around. Now, these dark dots that we see in the part that's not the tissue is either dirt on the glass or it could be pieces of the tissue that got smeared off on the glass, which might be why some of it looks like it's cells that have been stained. But this is actually not part of the glass. The way that this tissue is laid out, and this is all gray matter, the outer part doesn't pick up a whole lot of stain because it's not filled with a lot of cell bodies. It's mostly filled with dendrites of some of these cells. And then there's this dark kind of grainy looking part in here, which is filled with a bunch of little neurons. This kind of white streak down here in the deep part of the dark grainy looking stuff, that is white matter right there. But the gray matter on the outside, there are different regions that have different appearances because of how much that region is filled with cell bodies versus dendrites. Now, what we're going to look at is right where the two come together, where these dark spots are right at the edge. There's a little bit of a tear in the tissue right here. That's kind of unusual, but here it looks fairly intact. If I go to the 10x objective version here, there still looks like there's a little bit of a tear at the tissue here. But here's a couple of dark cells, another one there, another one there. And we can start to make out their nuclei a little bit. This is the same tissue at 40x, and here are those big cells, the nuclei that we can see, even a little better visualization of the nucleus there. These are really big cells that are right on the edge of this grainy looking area, and all those little grains in the tissue here, these are small neurons. Okay. But what we're interested in are these big neurons here, which are a specialized type of cell in the cerebellum. Okay. We only see the nucleus in the cytoplasm. We can see the hint of something maybe coming off here, and maybe coming off of this one here, maybe coming off there. But we can't really see any of the dendrites or axon coming off of either of these. Okay. The thing is these cells actually do have a huge dendritic tree. And we go back here, we can actually see the edge of the tissue right here. So all of this is that light staining part that I pointed out in the low power picture. It's filled with the dendrites of these cells here. Now these cells are spaced kind of far apart, so that suggests that the dendrites are pretty big, spreading trees, filling in all that extra space. And I'm not in the section of the book that shows it there, but that picture that had the 3 different types of multipolar neurons and the 1 bipolar neuron in the olfactory system, the middle picture in that was one of these cells. And they're called Purkinje cells. Now, Purkinje is the name of an anatomist who is rather famous for discovering a number of things. And the things he discovers usually get his name associated with them. There's not a better name for this type of cell, so we stick with the dead white guy name for it. But here's a number of pictures showing you what they look like. This picture here, for instance, is similar to what I was just showing you, and here's essentially the 10x objective, but somebody's drawn on it to highlight a little bit more what the cells look like. This picture, not a great one. This picture is stained with a fluorescent dye. Now, the fluorescent dye is injected into the cell, and it actually happens to be a molecule that associates with the lipids in the membrane. So it coats the entire cell, all of the cell membrane. And then when it's excited with UV radiation, UV light, it fluoresces this bright green. So we can kind of see the entire cell. We can see this dendrite coming off and all the little branches extending away from it. This is another micrograph of the same sort of thing. We can see these 2 rather extensive green cells. There's a lot of red cells sort of in the background. This has been stained with a couple of different things, so we can see a bunch of stuff going on. Here's another one that's pretty extensively stained. This is another one. These cells are known for their huge dendritic trees. And again, they need to be big cells to support all of those dendrites. Instead of the axons I was talking about before, it's still the same kind of idea. Okay. The other type of cell that was in that picture, so there are the olfactory cells and the Purkinje cell in the middle. The other cell that was in that picture is called a pyramidal cell. We don't have a good slide to show you that, so I'm just going to pull up some micrographs of it. This is one that's been stained with a heavy metal stain, usually a silver-based stain, which instead of glowing green like the fluorescent thing, it just turns black. But we can see a lot of the cell. They're called pyramidal because the cell body looks like a pyramid, which in 2 dimensions would be a triangle. The top of the pyramid has a dendrite that goes straight up, and then the base of the pyramid has a bunch of little dendrites sticking off and 1 axon going away from that. Here's a schematic representation of what this kind of cell looks like. Here's a number of different micrographs of pyramidal cells from different parts of the brain. Just some variety of what they look like, but there's a bunch of different ones. Here's another set of drawings of these types of cells showing some variety of their shape. But again, these are all multipolar cells. The point being that multipolar cells come in a lot of different shapes, but they're all collectively called multipolar cells because they have more than 2 things sticking off of them. So the book mentions pyramidal cells. We just don't have a good slide of it in our slide set, so I just wanted to show you some pictures here. Now, the other stuff I want to show you is about glial cells. I actually got a little bit ahead of myself and showed you one earlier, but let me go back to the spinal cord. Whoops, wrong one. This very low power spinal cord, where we can see the hole in the middle. The dark line that's standing right around that hole, those are ependymal cells. Okay. And I actually have a 10x objective picture of that part of the cell, that part of the slide. So you can see these cells, they kind of look like an epithelium. They're not. It's nervous tissue, but ependymal cells serve some functions that epithelial tissues serve elsewhere in the body. So we don't have any epithelial cells in the brain or spinal cord itself. Okay. Now, the cells that we see in the white matter and the smaller cells that we see in the gray matter are probably the other types of glial cells. We cannot, from this information, tell what type of cells they are. Most likely we have oligodendrocytes in the white matter because that's what's making the myelin, the insulation that is the white part of the white matter. There's probably a lot of that in the white matter. There might be some oligodendrocytes in the gray matter, but they're going to be much fewer because the more oligodendrocytes you have, the more white the matter is. So there's going to be very few if they're at all present in the gray matter. Instead, most of what we see in the gray matter is probably astrocytes. There may be a microglial cell or two somewhere scattered around, but there's no way to tell. And if the name isn't obvious enough, microglial cells are very small glial cells. So they're hard to pick out. But that's about what we can see of the cells, glial cells in this tissue here. Ependymal cells around the empty space in the middle of the tissue. Oligodendrocytes are probably all over the place in white matter. Astrocytes are probably all over the place in gray matter. Microglial cells, if they're present, are few and far between and very hard to pick out in the microscope. Okay. If we go back to the dorsal root ganglion, actually, let me go back to the one that's labeled unipolar cell. I already said this, but the nuclei that we see surrounding this neuron, those are the satellite cells. And they're named satellite cells because they look like they're orbiting this bigger cell in the center. Okay. Just beyond the neuron that we see here, there's a little bit of fibrous area here, which is probably axons passing along through the ganglion. And the neurons that we can see in here, the nuclei that we can see here would be Schwann cells that are insulating these axons. Okay. Not that we can see any detail to this. Now, to really see the myelin around axons, we don't want to use a cell stain like this. Cell stains react with charged particles like RNA and DNA and those sorts of things, and we can see that in the cells. Uncharged particles, which would be lipids for the most part, are not visible in stained tissue like this. I have a 40x objective view of the white matter, and I took this picture to point out we can see a dark spot in the center of this void of white around it, which shows us the axon, but we don't stain the myelin around it. Okay, so in here, what looks like these blank halos around little dark spots, that's the myelination. Okay, just we're staining for charged particles in this tissue, so we don't see the lipids because lipids have no charge to them. So if we want to see that stuff, we need to do something different, which is done with this in this slide here. Now, the pictures I showed you a little bit ago of the Purkinje cells and pyramidal cells are actually using a lipid-based stain. For the Purkinje cells, the one that fluoresced green, that stain was attracted to lipids, but we saw it because of fluorescent green. So it's just a weird kind of special kind of stain. In a couple of the pyramidal cell pictures I showed you, we saw the whole cell and it was black. Now, in some of them, it's because somebody drew them. They looked at them under the microscope and did a drawing of it, so it's black because the pen ink is black. But at least one of them, we were looking at a micrograph. We were looking at a cell with stain, and I actually said it was stained with a silver-based stain. Okay. silver is a heavy metal, and it can be used in that kind of staining to see the individual neurons. When we want to see lipids, we use a different type of heavy metal stain. We use what's called osmic acid. Osmic acid is referring to the heavy metal osmium. And you don't need to know anything about osmium other than it's a heavy metal. Referring to it as a heavy metal is important because heavy metals are heavy because the atomic nucleus, another use of the word nucleus in this course, has a lot of protons and neutrons. Most of the atoms we deal with have small nuclei. Oxygen has 8 protons, probably 8 neutrons. 16 particles in its nucleus. I don't remember the atomic number for osmium offhand, but let's just say it has 40, 60 some odd protons in it. It's probably going to have an equal number of neutrons. So it's a huge nucleus. Heavy metal. That's what heavy metal is getting. Not that it likes to turn the guitar amps up real loud or anything like that. Now, because the osmium is attracted to the lipid, when we shine light through it, the light is blocked anywhere where the osmium is present, and so it casts a shadow there. And that's basically what we're looking at. Now, the nervous tissue that we're looking at here is a nerve. Up at the top, the thing that kind of looks like a watermelon slice, and that's how a lot of students have described it, so that's how I refer to it. That's the nerve. Aside from that, the rest of what's in this slide is not nervous tissue. Now, it's a nerve, which by definition means it's in the periphery. If I called it a tract, we'd be in the central nervous system. And if it was a tract, it would be surrounded by more nervous tissue, because the central nervous system is only nervous tissue. But in the periphery, the nervous tissue exists side by side with other tissues. So what we're looking at here is a nerve that's surrounded by other tissue. Now, based on what I've told you about the staining here, what type of tissue is this nerve surrounded by? Why do you say that?
Because it's dark. And you said that heavy metal is attracted to lipids.
Right. So everywhere we see a big black dot there has to be lipids. Okay. The adipose tissue you looked at before using the cell stain, the lipid didn't pick up any stain. We saw the very thin cytoplasm or nuclei that are associated with adipose sites. Here, we're kind of looking at the reverse image. So surrounding these dark spots, there's kind of blank space because that's what the rest of the cell around you can't really see. I'm not going to look at that in too much detail because that's not terribly important. I want to look, however, at the slide in a bit more detail. Again, it's been stained with osmic acid. And so we're only seeing the lipid. And what we're seeing is the myelin surrounding the axon. So earlier when I was talking about myelination and I showed you an electron micrograph of an axon, that's sort of what we're seeing here. Now, unfortunately, we don't see a whole lot of detail here. This is only a 10x objective magnification. I didn't take a 40x because I didn't want to get into that much detail. But you can see a few things here that are dark bands surrounding a little vacant spot in the middle. Okay. The electron micrograph in the book that I showed you earlier, and I pointed out, you can see the layers of cell membrane that are wrapped around the axon. That's stained with osmic acid also. Osmic acid is one of the stains you use traditionally in electron micrographs. We're just using it here with light micrograph, but essentially we're seeing the same thing. Now we see more detail in the electron micrograph for a couple of reasons. One, electron micrographs give us more detail, and two, there's 2 stains in that thing besides the osmic acid. This is only stained with osmic acid. So we don't see anything inside the little void of this axon. Now, we can see some cracks in the tissue here. Nerves are actually organized very similarly to muscles. Remember when we learned about muscles, there was the epimysium that surrounds the whole muscle. There's a paramycium that surrounds a fascicle, and the endomycium that surrounds individual fibers. In a nerve, we have epineurium surrounding the entire nerve. We have perineurium surrounding fascicles or bundles of nerve fibers, and endoneurium surrounding individual nerves themselves, nerve axons themselves. At one time, people thought that nervous tissue and muscle tissue was very similar. And actually what we're going to get into the rest of today points at a little bit why that misconception was held. They behave very similarly electrically. So people thought that they were similar tissues, and so they were described as being organized in a very similar way. That's why epimycium, epineurium are kind of the same construction of the word. Let's see. So I don't have a 40x objective version of that. I do have the 40x objective of the white matter, which I showed you a little bit ago. Imagine this in negative. So the white space that we see here, if that were black, and the stained part in the middle, if that was white, that would essentially be what a 40x of that watermelon slice slide looks like. Okay. I also want to show you this picture here. This is also stained with osmic acid, but this is what's referred to as a teased nerve. So the picture we were just looking at before, the nerve was cut in half, okay, along its length. And then it was sectioned and cross-sectioned. So we saw the slices. If we looked at all of the slides of that particular thing, we just see sequential slices through that half of a nerve. Here, the nerve has been cut along its length also to open it up, to cut through the epineurium. And then forceps were used to pull the individual nerve fibers apart, break apart the fascicles, so we can see the individual nerves. Now, probably these fibers that are all kind of together were probably part of one fascicle, and these were probably part of another fascicle, and these were probably part of another fascicle. So the fascicles are a little easier to separate than the individual nerves, but with some work, the individual nerves could be pulled apart. This is still stained with osmic acid, so we're still seeing the myelin stain. It's just we're not seeing a cross-section of the nerve, we're looking at the length of the nerve. And what I would like to point about this is we can see a little gap right there, okay? And if we follow along, this thing here is a little crushed piece of tissue, so ignore that. So starting here, we follow this fiber along, and we don't see another little gap until we get down here, okay? Those gaps are important. They're called nodes of Ranvier, named after a dead French guy, so Ranvier, not Ranvier. Nodes of Ranvier. Those gaps are important, and the pictures of the myelin in the textbook that we saw, they were little packets, sheaths, that had little gaps separating. That's what we're looking at here. Those gaps are very important in how electrical information travels down the fiber. But I want to show you this because in a textbook, it's always going to misrepresent the distance between those nodes. And actually, let me. So here, this picture we started looking at, the myelin sheaths look fairly short, okay? But in fact, they're really long. The distance from one to the next is going to be a lot longer than this picture suggests. For the sake of a picture in a book, a drawing like this, they have to be made short. If it was drawn to scale, given the diameter of that axon, one myelin sheath would go off the page or screen, or however you want to think about it. So to get several onto the page together, it has to be drawn in this kind of misleading fashion. But keep in mind that really they're pretty far apart. Okay? So from here to there is the length of one myelin sheath. Okay? If you think about, you know, the axons I was talking about, like the one going to my big toe that's a meter long or so, If the myelin sheaths were really that short, then there'd be a whole bunch of them going all the way down. Now, there's a good number going down. The distance from the one spot to the next is measured in millimeters, which are thousandths of a meter. It might be 100 millimeters or something like that. Might be 50. I can't really remember the length. But there's going to be a finite number of sheaths along the length that you can really kind of make out. The distance from one to the next has been optimized over years and years of adaptation. And I'll talk about that a little bit later on, but we see that here. So that shows you the different types of tissue. Okay? If you want to look at these slides yourself under the microscope, you can go use room 511 and look at them. They're in the yellow box. I think slides 2 and 5, I don't have the actual list of slides handy, are the two nerve pictures, the watermelon and then the tease nerve. You just saw that, showed you there. I think 2 is a section nerve and 5 is a tease nerve. And then 6, 7, and 8 are the other three that I showed you. 6 is the spinal cord motor neuron, first one I showed you. 7 would be the dorsal ganglion, the slide's actually labeled spinal ganglion, and then 8 would be the cerebellum. 9 is a slide that's supposed to show you pyramidal cells from the cerebrum, but they're really bad slides. I don't like to use them in class because it's hard to see the pyramidal shape in them. But if you want to look at them, you're welcome to. So that's basically where these all come from. I labeled them a little differently from what the slides say so that to highlight what these pictures are actually showing you here. And you can get to these. I just made sure that this link is here in this folder. Oh, the other thing I forgot to mention, in this part of the course, you have the 4 assigned parts to the assignments for this chapter. There's a 5th assignment here called the Excitable Membranes Overview. This will be questions about electrical activity stuff, which I'm about to get to talk to. But some of the questions are going to relate back to the muscle chapters because what we're going to talk about in the context of neurons applies to muscles too. So it's just another 3-question, 6 points apiece kind of thing. It's structured just like a part 4. When you go in here, you won't see it. Okay. When you open this up, you'll see nervous tissue part 4, and you won't see excitable membrane. I mean, you'll see nervous tissue part 1. You won't see anything else. You got to complete part 1 to go into part 2. You got to complete part 2 to go on to part 3. You got to complete part 3 to go on to part 4. You have to complete part 4 to go on to the overview assignment. The score doesn't make any difference. Whatever score you get will allow you to go on to to the overview assignment, which really means that I don't have to grade it for you to go on. So once you complete part 4, that 5th assignment will be present. I have yet to grade part 4, but if you had to wait for me to grade part 4 to do that assignment, you'd probably be doing it the last day of the semester. Hopefully not, but I don't want to put you in that position. So you'll be able to do that assignment as soon as you complete part 4, even though part 4 has not been graded. And you only have one attempt on part 4, so if it was dependent on you getting 80% correct and going on to the next one, if you didn't get 80% correct in one shot, then you'd be out of luck. So part 4 just needs to be complete, and then you'll see the excitable membranes overview assignment there. So, speaking of excitable membranes, we need to go there. Now, unfortunately, we don't have a whole lot of time left in the class today. And I had actually kind of hoped to finish up all that stuff before we took a break, looking at the rest of the slides. Didn't happen. An hour really isn't a good enough amount of time to go through this. So I I'm going to try to impart to you an understanding of what's going on here as much as possible, and we might have to revisit it next week. Okay? As far as the assignments that are required here, if you're unsure of anything, please don't submit anything until you've asked me questions or we've had a chance to kind of regroup about this. But we're going to try to deal with this. Now, and this is just the nature of this being a summer course. So, this section I refer to as excitable membranes, because really it's more than just neurons being electrically active. It also has to do with muscle cells being active. But it's about membranes. Okay. Now remember, membranes are phospholipid bilayers. Okay. Things cannot cross through the phospholipid bilayer unless there's something that allows it to go across. Now, lipids actually can go across lipid bilayers all they want. They're freely diffusible, but we don't care about lipids here. We care about ions. So the electricity that's making this light up here come on is based on electrons. And electrons have a charge. The change in charge in the circuit that's connected to these lights is what makes the light light up. For cells, electrical activity is based on a change of charge across the membrane. And that charge is represented by the distribution of ions. Now, the ions we're most interested in are sodium and potassium. If you look at a periodic table, you'll notice that sodium and potassium are right next to each other in that first column. Okay? So the first row is hydrogen. It's got 1 proton and 1 electron. The second row, the first position, is lithium. It's got 3 protons, 3 electrons. 2 in its inner shell and 1 in its outer shell. The next row down, you have sodium, which has 11 protons and therefore 11 electrons. 2 in its inner shell, 8 in its second shell, and 1 in its outer shell. Okay. And then you go down from that and you have potassium, which has 19 protons, 2 in its inner shell, 8 in its second shell, 8 in its third shell, and 1 in its outer shell. What's the same in all 4 of those examples I just mentioned? There's only 1 electron in the outer shell of all of those. An ion of any of those just loses that 1 electron in its outer shell. So there's 1 less electron than there are protons in an ion. Which means that those ions will all have what charge? 1 more proton than electron. A single positive charge. Hydrogen ions are important, but not today. Okay. We'll see hydrogen ions in a lot of other places in this course. Lithium ions we don't care about. They're great for lithium ion batteries or medicine for bipolar disorder or something like that. But we don't really care about lithium. Then we have sodium and potassium. And they are both positively charged ions. Okay? Now, what we're interested in is a distribution of charge across the membrane. We can't get a distribution of charge between 2 equally charged ions. Okay? So what's important about sodium and potassium is they are the positive end of the spectrum that we're interested in. Okay? The negative end of the spectrum is are other ions. Okay? And the ions that we're mostly interested in there are proteins or phosphates. Okay? The thing about proteins and phosphates is they're big ions. They don't cross the cell membrane easily, or at all, for that matter. They're mostly stuck inside the cell. Okay? So we have a lot of negatively charged things inside the cell, and then we have positive ions that we can manipulate across the cell membrane that'll change things. Sodium is found more outside the cell than inside the cell. And in fact, there are 10 sodium ions outside the cell for every sodium ion inside the cell. Huge concentration difference in sodium. Potassium is more likely to be found inside the cell than outside, but its concentration gradient isn't quite as big. There's 5 or 6 potassiums inside for everyone outside. Not a big thing. So really we're talking about more positive charge outside the cell than inside the cell, and a lot of negative charge inside the cell that doesn't get out. Now, usually when I'm describing this, I like to hopefully get you to understand the idea of distribution charge by talking about one of these. Battery. The membrane essentially is a battery as far as this is concerned. There's positive charge outside the cell and negative charge inside the cell. Just like the battery has distributed across it, positive to negative charge. Now, I'm not a physicist. I'm not really great at electromagnetic physics or anything like that. So if I'm misdescribing this at all, I'm sorry, but I'm simplifying things as much as possible. Now, how much charge is distributed across this battery? Nobody ever knows the answer to this. Actually, one person in my career here has ever answered my question. But that's just because nobody pays attention. It says right on the battery how much charge it has. It says right there. Everybody read it? Okay. If I asked that question instead with this, you would have been able to tell me quickly. How much charge should 9-volt battery obviously has 9 volts in charge. No, it's 1.5.
Okay.
AA and AAA batteries are both 1.5 volt batteries. And if you look right there on the copper top end of this, it says AA, it has some other codes for what type of battery it is. It says 1.5 volts. Look at a AAA battery, you'll see the same thing. Now I use a AA battery because it has a positive and opposite, a negative pole. The 9-volt battery isn't a very good one to describe the membrane because the positive and negative poles are both on the same side of the cell. But everybody knows what voltage is. Now, charge is measured in volts. And how much is distributed across that battery is 1.5 volts, 1.5 volts. We're talking about the charge distributed across the membrane. We measure that in millivolts. Just like a millimeter is 1/1000th of a meter, a millivolt is 1/1000th of a volt. The charge that we measure across membranes is in the neighborhood of, the lowest charge we're going to deal with is going to be a little less than -70 millivolts, and the highest charge is going to be a little bit more, or is going to be in the neighborhood of 30 millivolts. For a change of about 100 millivolts. The difference between -70 and 30 is 100. -70 down here, 30 up here, even out. So 100 millivolt charge distributed can be discharged. 100 millivolts can be discharged across membrane. 1500 millivolts can be discharged from one of these 1.5 volt batteries. Just to give you a sense of scale. The charge that we're talking about in the cell is pretty small. Now, when a cell's not doing anything, when the cell's at rest, that charge is referred to as its resting potential. Potential is a word we use to describe that distribution of charge. And when we can allow that charge to change, when it moves, when ions move across the membrane, that's when we're going to see a change in the electrical distribution. Go from -70 to +30. At rest, the charge that we measure across the membrane is -70 millivolts. Now, that's not actually an absolute number. It's just a relative number. Relatively speaking, it's 70 millivolts more negative inside the cell than outside. That's assuming outside the cell is 0. Inside is negative 70. It could be outside the cell is 120. Inside the cell would be 50, negative 50. It's a relative difference. That's all I'm trying to know. Being relative isn't important, but just it's important. I do want to state that fact there. So this figure in the book here is just showing you how you measure volt. Okay. There's an electrode that pierces through the cell membrane and records what the electrical charge is inside, and then there's another electrode that's outside the cell. And you compare those two together, and you get negative 70 as the charge inside the cell. When we want to change that charge, when we want something to happen, then we need some sort of channel to allow things to go through. Proteins in the membrane have openings that allow ions to pass across the membrane. And by controlling those channels, we can see some changes happening across the membrane. Now, I could sit here and keep talking about this for a while, but I actually would rather show this to you. Maybe I saved this. Hold on a second. I'm looking to see if I have the. Not here. Okay. So, I don't have this saved in the video content section of Blackboard for you, but I have it here. And this is just, I have saved so I don't have to search through YouTube again. But this is a YouTube video. If you would like to watch it on your own, its title is Nerve Impulse Mechanism. So you can just go to YouTube and search for nerve impulse mechanism and you'll find it. Okay. Let me play this.
Need to hang a heavy mirror or picture? Use these helpful tips to hang it safely.
Okay, it has nothing to do with Lowe's, obviously.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell. making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
Okay. So let me go through the video a little bit and point out some things. I like this video. It does a really good job of explaining things and putting it in terms that I think work well for this course. First off, it illustrates the bigger picture of what's happening here with this example of touching a hot pan on the stove. Now in the book, actually, I skipped over in the interest of time, but I do kind of set up the same idea. There are several neurons that are part of processing information. The example I use in the book is about testing the temperature of water in a shower, but still it's the same idea. I use the shower thing because it involves higher processing a little bit, so it involves the brain too. Withdrawing your hand from a hot stove is a very reflexive action and just involves this basic input. And in fact, in this explanation here, the voiceover mentions this inner neuron. Actually, if you're touching something hot on the stove and you withdraw your hand that quickly, there's only two neurons involved. There's a sensory neuron that immediately tells the motor neuron to cause a contraction of the muscle. There isn't any inner neuron. An inner neuron would just slow the signal down by milliseconds, but enough to make the damage to the tissue potentially worse. So in the simplest reflex that this is suggesting, the pain receptors would immediately tell this neuron to cause contraction of the muscle and pull away. But there are situations where inner neurons are involved here. Okay, so that's fine. Now in this video and in many sources like this, when they're representing an electrical signal traveling, we saw this in the video last week, they're going to show it as a bright spot moving along the cell. Like this. Okay. That movement, that electrical signal, is what we call an action potential. Okay. The membrane's charge is referred to its potential, in sort of in the sense of potential energy. It's a battery. It's stored up some charge. When that charge is being discharged, something's happening. That's an action potential. That's the signal. Let's see. Then it zoomed in to look at what's going on at the membrane level. One of the things I really like about this video is that the entire time it's showing you the membrane, this thing here is chugging away. This is a sodium-potassium pump. 3 sodium ions are pulled from inside the cell and pushed out. And then 2 potassium ions are pulled from the outside and pumped in. It requires hydrolyzing ATP for this to happen. This is always happening, always keeping sodium out of the cell and forcing potassium into the cell. Okay. So it's kind of nice that it does that. Now, as she's saying this, and let me see if I'm not going to be able to find the exact frame with the voiceover down the bottom, but as she's saying this, she's saying there's sodium outside the cell and there's potassium inside the cell. And then she goes right to, and therefore there's a positive charge outside the cell. Okay. I set all this up by first saying that sodium and potassium have the same charge, because something that often gets overlooked in this is that they in fact do have the same charge. And distributing sodium and potassium across the membrane is not the basis of this charge differential. It's because inside the cell there are all these negatively charged proteins and phosphates that don't come out of the cell. And we can manipulate the charge, whether it's positive outside versus inside or positive inside versus outside, by moving positively charged ions across the cell. We need to use the concentration gradients, or electrochemical gradients, as the video says, of sodium potassium. And it has to be specific where they are when we have certain things happen, so that we can reproduce it over and over again. Now, for the sake of this video, they're representing the channels that allow these ions across using color coding. So the green ones are sodium channels and the purple ones are potassium channels. They do not look like these pictures. Okay? These are just schematic representations. The sodium ion channel actually has 2 parts to it. Okay? It has 2 gates, or 2 things that can open or close. There's the activation gate and the inactivation gate. Gates are responsive to voltage changes. We call these voltage gated channels. So the green one is the voltage gated sodium channel. The purple one is a voltage gated potassium channel. The potassium channel only has 1 gate. It's either open or it's closed. But the sodium one actually has 2 gates, so it opens and closes under different conditions, which we'll see in a second. Actually, I did catch the frame. So here's where voiceover is saying, established by the unequal distribution of sodium ions outside of the cell and potassium ions inside. They both have the same charge. Being distributed across the membrane does not affect the membrane charge. It's because other ions play a part and they have a negative charge. But that usually gets left out in this stuff. Okay, so here we see the glow representing a change in charge. We have positive inside versus negative outside. Remember, as this was happening, the glow moved across the screen from one side to the other. So we have the reverse charge here where that glow passed over. The glow hasn't gotten over here, so we still have positive outside and negative inside. Okay. It's when the sodium comes inside that it gets positive inside. Okay. And the potassium channels are not open yet. Okay. Here it talks about how potential changes at different places can sum together. A couple of positive charges changes that come together, then you'll have more of a positive change. And that'll lead to what's called the action potential that travels down the axon. Okay. This is labeled resting potential, but actually what's going on in the graph isn't technically resting potential. Resting potential is when the cell's at rest, nothing's happening. The membrane in this picture is in fact at rest. All of the channels are closed. There's a positive charge outside. There's a negative charge inside. If we measured the distribution of charge here, we would see it's at -70. Okay. But in the graph here, they're showing the line going up. It's going from -70 towards -55 is actually the value. And there'd have to be something moving across the membrane for us to see a change. We don't actually see that here. So a little mismatch in what they're talking about, but that's fine. What's important is once we get to that -55 value, then the sodium channels open. If you want to, there's a little, if you hit the volume thing down here at the bottom, that'll mute the video if you want to. Yeah. I sometimes forget to do that myself as I'm doing this, but anyways, so right now the sodium channels are open, sodium's coming through, the inside's getting positive compared to the outside. And when that happens, we call that depolarization. The membrane voltage is going from -55 up to +30. It's becoming less negative, which is another way of defining depolarizing. Depolarization, less negative. It's not exactly what depolarization means, but in this context, it makes sense. Okay. When we're becoming less negative, we're depolarizing the membrane. Okay. Now, the two gates I talked about in the sodium channel, which are not represented in this picture, one of them opens when we hit -55 millivolts. The other one closes when we hit +30. So that means this depolarization period, which is highlighted in the graph up here, is the only time that the sodium channels actually open. Okay. We have to get back almost to resting potential for things to be reset to happen again. And then, come on, there we go. Then the potassium channels open. Okay. And we go into what's called repolarization, which is the opposite of depolarization. So if depolarization is when things get less negative, repolarization is when things get negative again. And that happens because potassium channels open and the positive charge of potassium leaves the cell, pulling the positive charge out again. It's a different ion, but it's still the same charge. Now, potassium, voltage-gated potassium channels are much more simple than the voltage-gated sodium channels. They only have one thing that opens them, one charge. It's complicated, however, because they're slow. They open at, they're told to open at -50 millivolts, but they're slow. They don't actually open for about a half a millisecond after that charge is hit. So over here, we're at -55. During depolarization, the membrane goes to -50, and that tells the potassium channels to open. But it takes them about a half a millisecond to open up, and by that time, depolarization's hit its peak. Okay. So these open up right here and cause repolarization from the peak back down, actually because they're slow to open. As potassium leaves, the cell gets more and more negative, and it actually gets too negative. It goes past -70 millivolts, which is resting potential. And the way this graph is drawn, you can kind of see that the bottom of the graph is here, and it slowly comes back up to -70 there. Repolarization ends with it being too polarized. what we call hyperpolarized. And that's because the channels are slow to close, just like they're slow to open. Okay? They're told to open at negative 50 millivolts, but they don't actually open for a half a millisecond after. When we pass negative 50 again, going the other direction, they're told to close. But it takes them about a half a millisecond to get closed. So much potassium is left in that half a millisecond that it actually gets below negative 70, hyperpolarized. And then once that's closed, the sodium-potassium pump will pump sodium out, potassium back in, and we get back to the resting potential. Okay. That's essentially what the action potential is. At rest, the cell's at negative 70. Something happens to get it to negative 55, which activates the sodium channels. Sodium rushes in, and we depolarize very quickly till peak when they're inactivated. Inactivating sodium channels and the opening of potassium channels actually happens at the exact same time. They're synchronized. And so sodium stops rushing in, and now potassium starts to rush out, and positive charge coming in depolarized, positive charge going out repolarizes. And we go back down, but because they're slow to close, just like they were slow to open, they go too far, and it over polarizes. It gets past resting potential. And then after they finally close, it gets back to normal. Now, that hyperpolarization, while it sounds like a mistake, is actually an important feature. Once we've gotten past, during repolarization, we've gotten past negative 55 again, the sodium channels are ready to go. They're recycled, they're ready to go for another action potential. Between the beginning of depolarization and then that point repolarization that they're finally reset, you can't get another action potential to fire on. You can't open more sodium channels because they're all already open. If it's really hot in your house and you open all the windows trying to cool it off and it doesn't get cool enough, it doesn't cool off enough, you can't open more windows. All of them are open. You can't get more sodium into the cell to make more of an action potential. They all open up, and so the cell's just going to depolarize. Once they've reset, then you can open them again for another action potential. The thing is now the potassium things are open. And if sodium starts rushing in while potassium's rushing out, those two charges are going to kind of cancel each other out. You're not going to get an action potential. While one action potential is taking place, another one can't start, which is called the refractory period. There's two aspects to the refractory period. There's the absolute refractory period when sodium channels can't be opened again or open more. When sodium's already doing its thing or it's inactivated, you can't fire off another action potential. You can't increase the action potential that's in place. Once sodium channels are ready to go again, you still are in the refractory period because what potassium is doing, but we call that the relative refractory period. Because of potassium leaving, the membrane potential is headed to a negative number, below negative 70. You have to counteract that potassium charge to get it back up to positive 55. So even if not positive 55, negative 55. Even if after it passes negative 55 and at negative 53, you send a new signal in, if it's the same signal that started off before, it won't be strong enough to turn it around and get you back to 55. That same signal will just slow this down and it'll hit negative 70. It won't go past negative 70 probably, but it won't get to negative 55. During the relative refractory period, you can start a new signal, but it needs to be a stronger stimulus. So absolute refractory period, absolutely no new action potentials. Relative refractory period, relatively you can with a stronger stimulus. Get those two concepts? Trying to kind of go through this stuff quick because of the time price we have here. But that's refractory period. The video doesn't talk about that. I'm adding that in because we're talking about this aspect of the video. Now the video does go on after talking about the action potential. Here we go. Oh, no, before, it's still talking about the action potential here. I like this part of what the video says because a lot of sources don't make this point. The ions that are moving are not moving very far. This sodium channel is going to diffuse through to the side, making the inside positive. Or these potassium channels are going to move out. They're not moving, not like a potassium ion way over here is going to necessarily move out there. There's just so far that these ions have to move. And so it all happens very, very fast. The limiting factor is really how fast certain ion channels open. The sodium ion channels are pretty much instantaneous. The potassium ion channels are a little bit slow. And so all of that kind of works together to give us a very specific shape to the action potential curve. However, once the sodium gets in, even though it's only moved a little bit, it can diffuse up and down the membrane. And when it comes in here, as it diffuses down here, it'll then make it possible for the next channel to open. And sodium coming in here will diffuse down and make it possible for the next channel to open. So the sodium will move a little bit, but it only has to move across the membrane a very short distance for any changes to happen. Then it goes to talk about the synapse here, which is essentially the same thing as the neuromuscular junction that we talked about before. If it was a synapse with a muscle cell, it would be a neuromuscular junction. But in this picture, we're actually looking at a synapse between two neurons. There's a little gap between the two called a synaptic cleft. And then inside the end of the axon that the signal is traveling along, there are vesicles filled with chemicals called neurotransmitters. And those neurotransmitters will get released from the end of the axon through exocytosis, skipped over the frame that actually shows that happening. But these little pie pieces here represent neurotransmitters that have been released. And they bind to a spot on the receptor. So a protein in the target cell binds to the, we can kind of see it happening here, over here, binds to the neurotransmitter. And those receptors are channels. When the thing binds to it, they open up, and sodium can rush through. Now, when the video starts talking about it, and I point out there's a little mistake, it says resting potential while the curve is starting to go up to -55. And I said that's not actually resting potential. When that's changing, I'm going to see if I can get back to that spot. So this little change in the curve right here, where it's going from -70 to -55, this isn't really part of the action potential. This is stimulus that's getting everything started. And an example of that kind of stimulus would be what I was just showing you at the synapse. Come on. Okay. So when the neurotransmitter binds to its receptor and this channel opens and sodium rushes in, that's going to start depolarization. Okay. So as that starts, so this membrane, when those channels are closed, is at -70 millivolts. When it opens and sodium rushes through, then it's going to start to depolarize and get to -55. Once it hits -55, then the voltage-gated sodium channels will be activated and more sodium will rush in, and you'll get an action potential. oversimplifying things just a little bit there, but that's essentially what's happening. So hopefully that's a quick little trip through what's happening in the membrane. There are a couple of other things I want to mention, which I'll go back to the book for. So in the book, as I'm setting this stuff up, it's talking about channels. I'm not crazy about these drawings. The art editor and I went around about these a little bit. These are schematic drawings, cartoons, if you will, of these things, and so they're not necessarily going to be exactly correct representations. But here we have a closed channel, and here we have an open channel. This is a ligand-gated channel, which would be like a neurotransmitter receptor. So these red triangles are neurotransmitters. When they're attached to the channel opens up. This is a mechanically gated channel. Again, very schematic representation. I kind of think of it looking like a trash can with a lid. Okay, so normally the lid's closed, but if something mechanically happens to the cell, if it's squeezed, physically squeezed, the lid will pop up on the channel and then ions can move across. Either of those things, a chemical signal or a physical change in the cell, can open these channels. And those are two examples of how we get from the -70 to -55. Then when we're talking about activating sodium channels, we're going to be dealing with a voltage-gated channel. Now, this picture is not of the sodium channel. This is, if anything, a representation of the potassium channel, which has one gate. Sodium channel has two gates, a little more complicated. But they're at the under the control of voltage changes in the membrane. Okay, that's what that's getting. I don't have a link directly to it, and I didn't want to take the time to try to find it, but there's another type of channel called a leak channel. I really don't like the picture of it, but it can be closed or it can be open, but nothing controls when it opens and closes, so things can leak across. Leakage channels are important to getting the specific values of the charge of the membrane, but for understanding what's really going on, they're not that important to get into, so I'm going to jump over them. But understand that in the book, they're mentioned. And we see them. Now in the book, I introduce the graph for the action potential. When we have a flat line at -70, that's resting membrane potential. When we go up to -55, that's when something happens. Neurotransmitter binds to its receptor, or a mechanical channel opens, or whatever. Something depolarizes it to -55. -55 is when the sodium channels are activated. It's called threshold. And we get rapid depolarization. And then repolarization is when the potassium channels have finally opened. They're a little slow, remember. They're a little slow to close also, so it goes too far and hyperpolarizes before it gets back to resting potential. I have two versions of this picture. This one's kind of simple, just showing depolarization, repolarization, hyperpolarization. Three major events of an action potential. Down below, there's the same thing again with a few more labels to it. Kind of walks through what's happening as it's described in the, what do we call that? Figure caption. Propagation of the action potential, you can read through that. It's basically getting at the idea of how it moves down the length of an axon. And the next section about communication between neurons. First off, for electrical activity, there's another concept to talk about called a graded potential, which is when a change in the membrane charge is dependent on the size of the stimulus. Action potentials always behave the same way. Start at -70, they go up to +30. they go back down past -70, and then they get back to the resting potential. They always do the same thing. The numbers might vary from cell to cell a little bit, but for one cell, the action potential is always identical. But there are some situations where membrane potential changes are variable. We call those graded potentials. And the change is based on the size of the stimulus. A strong stimulus will make the membrane potential change a lot. A weak stimulus will not make it change very much at all. Now, it's really hard to demonstrate this idea based on single potentials. So it's not like I can say a strong stimulus like that is going to cause an action potential, and a weak stimulus like that is not. You could hear that, then it created action potentials in your system. But you can kind of imagine that how strong the stimulus is can relate to how much the membrane potential changes. So a lot of really light, ball claps, whatever you want to call them, add up to the same kind of signal that a single thunderclap can generate. In this figure that's kind of demonstrated, these different sized depolarizations represent different sized stimuli. There's 4 here, actually. The 2 in the middle are kind of the same height, but they can also be different in time. Okay, so a long, loud sound, a long, soft sound or a brief, loud sound can both end up being similar stimuli. It's also possible for the membrane to hyperpolarize. Okay, so some things can make it harder for the cell to generate action potentials. Okay. So we call those depolarizing or hyperpolarizing graded potentials. Graded potentials can happen in a lot of different ways. The most important types of graded potentials, right there, it's saying type of graded potential. The most important type of graded potential is a postsynaptic potential, or PSP. That's what happens in the target cell at the synapse. Now, it's most important because it's the one that we want to talk about the most, but it's not the most important necessarily because it's better than other ones. Graded potentials are happening elsewhere, but we're not going to talk about them in as much detail. Now, postsynaptic potentials can depolarize or hyperpolarize. They're graded potentials. When it's depolarization, we call it an excitatory postsynaptic potential because it has the likelihood of exciting the target neuron or target cell. When it's hyperpolarizing, we call it an inhibitory postsynaptic potential because it's inhibiting the cell from firing off, the target cell from firing off its own action potentials. We can summate these together. The video kind of alluded to that. But this picture, which I'm still not entirely convinced I like, but it turned out better than I thought, shows that graded potentials of different sizes, okay, and the green ones are excitatory, the purple ones are inhibitory, okay, and then the yellow line is just the membrane doing its own thing. So if we have a couple of excitatory ones, we'll build up the charge in the membrane, didn't quite reach -55, and an inhibitory one brings it back down. And then this little dip right here represents that there's no activity at the cell. So it's trying to get back to resting potential on its own. And then another excitatory and an inhibitory, another excitatory, and we finally hit threshold and we get the action potential. Okay. So this is just showing a number of events that can all kind of happen together. And if the net result is going from -70 to -55, then we generate an action potential. That's kind of what that's getting at. The book goes into explaining synapses in some detail, a little bullet list of the kind of things that you'd expect to see there. An explanation of what causes the chemical release at the end of the axon. The action potential getting here will cause the physical release of chemicals from that. And then neurotransmitter systems is just talking about the chemical classification of synapses. What neurotransmitters are involved are important for how they work. And this table kind of presents that. Now, let me try to explain this table because I want to just highlight the important things that you want to get out of it. In the table, each row is giving you some information about a particular set of neurotransmitters. Neurotransmitters usually can be thought of in two ways. Either they're the chemical class that they belong to, which is how this table is organized, or the specific neurotransmitter system that they're part of. Now the first column, so the second column in this table, at the top it says cholinergic. That's referring to the family of synapses where acetylcholine is active. Cholinergic refers to acetylcholine. There are two types of receptors that acetylcholine binds to. They're referred to as the nicotinic and muscarinic receptors. They're called that because they're activated coincidentally by a couple of drugs. You've probably heard of nicotine, but don't get caught up in what nicotine is actually at this point. You've probably not heard of muscarine. So nicotine can activate some acetylcholine receptors just like acetylcholine, but doesn't do anything to other ones. Muscarine activates those other ones just like acetylcholine, but doesn't do anything to the ones that nicotine can activate. So we have chemically defined classes of receptors that are all activated by acetylcholine. Now, the way that I've always described this is to use the key and lock metaphor for neurotransmitters and their receptors. Now, I used to have a way to show you this because I used to have keys that worked the locks in this department in the way I'm about to describe. But they rekeyed the locks and they ruined this example. But used to be that I had one key that opened this classroom door, and I had another key that opened the AV room door across the hall. And then I had a third key that opened my office door. My office door key was actually a master key for the department, which I got because the person that had that office before me had the master key. So I could open the classroom or AV room keys with the master key. I could open the classroom door with the classroom key, but I could not open the AV room door with the classroom key. I could open the AV room door with the AV room key, but I could not open the classroom with the AV room key. So we're working through this. The master key is like acetylcholine. It can open either door. The classroom key is like nicotine. It opens one door but not the other. The AV room key is like muscarine. It opens the other door but not the classroom. That's kind of the idea of the relationship between these molecules. That's just to say that there are two types of receptors that acetylcholine activates. And it's a slightly unfortunate coincidence that you probably know what nicotine is. Just pretend it's some random drug you've never heard of at this point. And then it talks about how we get rid of acetylcholine and what effects acetylcholine has. The nicotinic receptor is an ion channel, which we've seen before. Acetylcholine binds to it, the channel opens, sodium rushes in, which causes depolarization. Muscarinic receptors are actually a different type of receptor. They're not ion channels. They are activated and they can tell ion channels to open up. Most of the time, they tell ion channels to open up and allow sodium into the cell and depolarize it. But there are situations when there are muscarinic receptors that open channels that cause hyperpolarization. So for acetylcholine, it can do either depolarization or hyperpolarization. It can cause either excitatory postsynaptic potentials or inhibitory postsynaptic potentials. Depends on the exact receptor. The next column is called amino acids. These are amino acids that act as neurotransmitters. The most common neurotransmitters in the nervous system are glutamate and GABA. Glutamate is an amino acid that we use to make proteins in our body, and it also is used as a neurotransmitter. An enzyme can modify glutamate just a little bit into another molecule called GABA, which we call it GABA, that's all I'm going to tell you the name stands for. Don't worry about what GABA stands for. Nobody ever calls it by its full name, unless they're trying to show off. Glutamate has glutamate receptors. GABA has GABA receptors. They are different neurotransmitters. It's not like acetylcholine with two different receptors. Two different neurotransmitters, each with their own receptors. There are a family of glutamate receptors and there are a family of GABA receptors. But for our purposes, we're going to lump them all together. Glutamate receptors always cause depolarization. GABA receptors always cause hyperpolarization. Those are the two most common neurotransmitters in your central nervous system. Also, an amino acid neurotransmitter that I've got listed there is glycine. Glycine is also going to cause hyperpolarization, but it does it through its own set of channels. And in fact, I think glycine receptors are chloride channels, which would let negative charges into the cell, which would make the cell more negative than it already is, which is hyperpolarization. Glutamate is sometimes referred to as an excitatory amino acid or an excitatory neurotransmitter because it always excites the target cells. Glycine and GABA are referred to as inhibitory amino acids or inhibitory neurotransmitters because they always hyperpolarize their target cells. The next group are called biogenic amines, which are made from amino acids. That's what biogenic amine refers to. They are not amino acids. One of them is serotonin, which has a family of receptors, but all serotonin receptors cause hyperpolarization. It's also sometimes referred to as an inhibitory neurotransmitter. Then we have dopamine, norepinephrine, and then in parentheses I have epinephrine. Those three chemicals are actually related to each other. Dopamine is made from an amino acid called tyrosine. Norepinephrine is made from dopamine, and epinephrine is made from norepinephrine. Dopamine has its own family of receptors. They're classified as D1 or D2 receptors. D1 receptors, if I remember this correctly, are excitatory and D2 receptors are inhibitory. 50% chance I have that backwards, but I'm pretty sure I have that right. Doesn't matter if I have it backwards. One's excitatory and others inhibitory. Dopamine can do either thing, depending on the receptor. Norepinephrine and epinephrine, which isn't really a neurotransmitter, bind to their own class of receptors. They both bind to the same receptors. They're referred to as alpha receptors and beta receptors. Some alpha receptors and some beta receptors cause hyperpolarization. Some alpha receptors or some beta receptors cause depolarization, which is to say norepinephrine and epinephrine can cause either excitation or inhibition. So acetylcholine, dopamine, norepinephrine / epinephrine, depending on the receptor, can cause either excitation or inhibition. Glutamate, glycine, GABA, serotonin cause only one or the other. Glutamate always causes excitation. Glycine, GABA, and serotonin always cause inhibition because all of their receptors do the same thing. The last column there says neuropeptides. I'm actually not going to talk about neuropeptides, and in fact, never do. I put it in the book because some people like to cover that. But, I don't deal with neuropeptides. It's just a little bit more confusing stuff that we don't want to deal with. And if you aren't confused yet, well, one, you're going to do great in this. So that kind of wraps up talking about the synapses and neurotransmitters. Now, I have technically gotten through all the material. If you have absorbed all the materials, it's a completely different story. I have satisfied my plans to cover all this material. In the coming week, as you're working on this chapter's assignments, if you run across stuff that's just, you don't understand, feel free to contact me, feel free to not submit stuff and commit to a score on an assignment until next week or something like that. These assignments are technically due Monday night next week, but I would rather you submit them late and get good grades on them because we have a chance to talk about it next Tuesday, rather than you say, oh my God, I gotta get all done by the night. Put it in, get bad grades, and ruin things for the rest of the semester. So, work on them, ask questions by text or email as you come across them. Feel free to research things far and wide. There are other videos on YouTube that explain all of this in more detail, but I didn't show them all. The one I did show you, I kind of like. And again, remember, if you want to watch that again, it's called Nerve Impulse Mechanism. Just search for that on YouTube and you can watch that again. And there's a number of videos that come up with that explain other aspects of the nervous system. So, there you go. Say that again?
The lab quiz you do?
No, don't do the lab quiz. It's not actually in this week's folder. It's in next week's folder because we got more lab stuff to do next week. Actually, you'll have 2 lab quizzes next week.
Can I ask you a question?
Sure.
Since we're on the topic, just so I can understand.